We have been asking the wrong question about this animal for 250 years. Since the Italian priest Lazzaro Spallanzani first described them in 1776, tiny creatures he called il tardigrado, the slow stepper, scientists have framed the tardigrade as a survivor. The ultimate survivor, the unkillable, the indestructible, the animal that laughs at conditions that would kill everything else. Here is the problem with that framing. It is not what happened. The tardigrade did not evolve to survive the vacuum of space. It did not develop its toolkit of molecular protections in order to walk away from nuclear radiation doses that would flatten a human cell culture in minutes. It did not engineer its biochemistry with any of those scenarios in mind, because evolution does not engineer anything with any scenario in mind. Evolution is not planning. Evolution is accounting. The tardigrade's invincibility is a side effect, a byproduct of solving a completely different problem, in a completely mundane environment, 500 million years ago. And understanding what that original problem actually was changes everything about how we read this animal, and everything about what its existence might eventually do for ours. The tardigrade is not an extremophile. This is a technical distinction that matters enormously and gets almost no popular attention. An extremophile is an organism that loves extreme conditions, that has evolved to specifically thrive where others cannot. Certain archaea live only inside volcanic hydrothermal vents. Certain bacteria grow optimally in pH levels that would dissolve metal. They need the extreme. Remove it, and they die. The tardigrade needs none of this. Most species prefer films of water on mosses, lichens, and leaf litter. They are, in their default state, perfectly ordinary microscopic animals going about the unremarkable business of eating, moving, and reproducing in damp vegetation. They are found on every continent on Earth, from the poles to the equator, from mountain peaks to ocean floors, because they can tolerate every environment, not because they prefer any particular extreme. The technical term for this is extremotolerance, not extremophily. The difference is the difference between a soldier who happens to know how to swim versus a fish. And the question that biology has been slowly, carefully answering over the past 30 years is, how exactly did an animal that lives in wet moss develop a biochemical toolkit capable of surviving the vacuum of space? What was it actually solving? And why did the solution end up being so catastrophically over-engineered? Before we look at the chemistry, we need to look at the body, because the body is its own paradox. Tardigrades are bilaterally symmetrical animals with 8 legs, a distinct head region, a complete digestive tract with a mouth, pharynx, esophagus, midgut, and rectum, a rudimentary nervous system with a brain ganglion and ventral nerve cord, and sensory organs, including eye spots in many species. They have muscles. They have claws. Some species have what appear to be internal gonads. They are, in the full biological sense, animals, not bacteria, not fungi, not some intermediate category of biological organization. They are also, in many species, approximately 0.1 to 1.5 millimeters long. Here is what is strange about this. An adult tardigrade has a fixed number of cells, a phenomenon called utily. Depending on species, this number runs between approximately 800 and 40,000. It does not grow through cell division as an adult. Instead, individual cells expand. The organism reaches its final cell count and then simply gets bigger by inflating what it already has. The implications of utily for cancer biology are worth dwelling on. Cancer is, at its core, uncontrolled cell division. Cells that refuse to stop replicating. In an organism that does not divide its somatic cells after reaching maturity, The primary mechanism by which cancer originates is eliminated. The tardigrade is not immune to all cellular damage. But the specific failure mode that kills more humans than almost anything else is, architecturally, not available to it. This was not planned. It is simply what happens when an animal is small enough that the metabolic cost of maintaining cell division machinery outweighs the benefit. Uteli is an energy-saving measure that happened to produce cancer resistance as a structural consequence. And then there is the walking. A tardigrade navigates terrain using a gait that biomechanicists have confirmed is functionally identical to the locomotion of much larger arthropods. The same alternating tripod pattern used by stick insects, which are approximately 500,000 times larger. It always keeps at least 3 legs on the substrate for stability. It corrects for slope and surface texture. It navigates the microarchitecture of moss leaves as though it were crossing complex terrain, because, at its scale, it is. An organism built at the lower boundary of animal complexity, operating with a nervous system that can be counted in neurons, uses the same optimized locomotion strategy as insects that evolved independently, at vastly larger scale. This is convergent engineering. 2 completely separate evolutionary paths arriving at the same mechanical solution, because, at the level of physics, it is simply the best answer. When a tardigrade's environment begins to dry out, it does something that has no parallel in vertebrate biology. It shuts itself down. Not slowly, not partially, completely. The process is called anhydrobiosis, life without water. And it produces the ton state, a contracted, dehydrated barrel in which metabolic activity drops to approximately 0.01% of normal. Measurable respiration essentially ceases. All cellular processes that require water as a solvent, which is to say, essentially all cellular processes, pause. The animal is not dead. It is not sleeping. It is in a state for which English does not have a precise word, because no human experience corresponds to it. Something between suspension and absence. The question that occupied biologists for decades was, how does a cell survive complete dehydration? Every biological membrane we understand depends on water. Every protein we know of requires hydration to maintain its functional shape. Remove water from a cell, and membranes collapse. Proteins unfold and aggregate. The molecular machinery of life ceases. Or it does in every organism except the tardigrade. The answer, identified in a landmark 2017 paper in Molecular Cell, turned out to involve a family of proteins so unusual that the field coined a new category for them: cytoplasmic abundant heat soluble proteins, known as CAHS proteins, which belong to the broader class of intrinsically disordered proteins. Unlike conventional proteins, which fold into specific 3-dimensional shapes to perform specific functions, CAHS proteins have no fixed structure in their hydrated state. They are chemically disordered, flexible, shape-shifting. When water leaves the cell, CAHS proteins do something that no engineering analog quite captures. They vitrify, transitioning from a disordered liquid state into a non-crystalline amorphous solid, a biological glass, not a crystal, which would introduce sharp edges and fracture points. A glass, a continuous, uniform, extremely viscous material that physically embeds every cellular component, every membrane, every organelle, every protein, in a protective matrix so dense that molecular motion essentially stops. The cell does not collapse when the water leaves. It is held in place by the glass the tardigrade made from its own proteins. Every fragile structure is suspended exactly where it was, maintained in its functional geometry, waiting for water to return. A 2025 study in protein science extended this understanding further, demonstrating that different phases of CAHS proteins, liquid solution versus gel, protect different types of cellular machinery during different stages of drying. The system is not a single protective mechanism. It is a layered, adaptive response that modulates its own physical properties depending on which part of the desiccation process the cell is currently in. This is not a simple stress response. This is a precision-engineered shutdown protocol running in an animal with a brain the size of a pinhead. Radiation kills by breaking DNA. High-energy photons and particles, X-rays, gamma rays, cosmic rays, collide with the tightly coiled chromosomes inside a cell nucleus and snap the phosphodiester backbone. A sufficient dose produces enough double-strand breaks simultaneously that the cell's repair machinery is overwhelmed. The chromosome fragments. The cell's ability to replicate itself is destroyed. At sufficient exposure, every cell in the body is affected at once. This is why radiation therapy works against tumors. It destroys DNA faster than any cell can repair it. A tardigrade can survive radiation doses of up to 5,000 gray, a unit of absorbed radiation dose. The lethal dose for a human being is approximately 5 gray. The tardigrade survives exposure 1,000 times greater. In 2016, researchers at the University of Tokyo published the high-quality genome of Ramazotius variornatus, one of the most radiation-tolerant tardigrade species, and found, embedded in that genome, a gene encoding a protein they named Dsup, damage suppressor. Dsup had no known counterpart in any other animal on Earth. It was, in the truest biological sense, unique. When they expressed Dsup in human-cultured cells and exposed those cells to X-ray radiation, DNA damage was reduced by approximately 40% compared to control cells without the protein. When they exposed tardigrades engineered to lack Dsup, their radiation tolerance dropped significantly. The protein was not a byproduct. It was a mechanism. What Dsup does, at the molecular level, is physically wrap around chromatin, the complex of DNA and histone proteins that makes up chromosomes. It is intrinsically disordered, like the CAHS proteins, which means it can conform to the irregular surface of nucleosomal DNA. It embeds itself across the genome, not at specific sites, but distributed broadly, and it physically intercepts the reactive oxygen species that radiation generates before those molecules can reach the DNA backbone. It is, in the language of engineering, a distributed armor layer. A molecular bumper fitted to every chromosome. In September 2025, a paper in Nature Communications demonstrated that when Dsup is expressed in yeast, an entirely different kingdom of life, it reduces oxidative DNA damage and extends yeast lifespan under chronic oxidative stress. The protein works across evolutionary distances that separate the tardigrade from yeast by over 1 billion years of divergence. And in February 2025, MIT researchers published results in Nature Biomedical Engineering showing that Dsup mRNA, delivered via lipid polymer nanoparticles to mouse mucosal tissues before radiation exposure, reduced radiation-induced DNA damage in those tissues by approximately 50%, without affecting the tumor's response to radiation. A protein that evolved in a half-millimeter animal living in roof moss is now being engineered into a potential clinical tool for protecting cancer patients from the side effects of radiotherapy. The tardigrade did not evolve Dsup to protect cancer patients. It evolved Dsup because its environment, the surface of damp mosses, exposed to sunlight and desiccation, involves significant oxidative stress, and oxidative stress damages DNA. The radiation protection is, again, a side effect. a piece of molecular machinery built for one context that works with extraordinary efficiency in a context the tardigrade has never encountered and never will. Strip away the remarkable survival chemistry and look at what a tardigrade actually does with its days, and you find an animal of complete ordinariness. It eats, it moves, it reproduces. It eats again. Its mouth parts are a pair of hardened stylets, needle-like lancets made of the protein chitin, flanking a circular oral opening connected to a muscular pump-like pharynx. The stylets pierce the cell walls of algae, moss cells, and plant tissue. The pharynx creates suction, pulling the cellular contents out through the breach. It is simple, mechanical, efficient, and completely unspectacular. Some marine tardigrade species have added to this basic toolkit in ways that are harder to describe without reaching for comparisons. Certain species carry appendages that can only be described in the vocabulary of Dr. Seuss. Flagpole-shaped tail projections, structures that function like inflatable anchors, expanding to attach to a substrate and deflating to release, and sensory barbels that superficially resemble nothing so much as decorative antennae on an animal too small to see with the naked eye. These are not adaptations for extreme environments. They are adaptations for the particular microstructure of marine sediment, for navigating the spaces between sand grains, for anchoring against water currents that, at tardigrade scale, are equivalent to hurricane-force winds. The extremotolerance that makes headlines is built on a foundation of completely pragmatic, completely mundane solutions to the engineering problems of being very small in a wet world. To understand why the tardigrade has the survival toolkit it has, you need to go back approximately 530 million years to the Cambrian seas before tardigrades had colonized land, before the mosses and lichens they now inhabit had evolved. The ancestral tardigrade problem was not radiation. It was not vacuum. It was osmosis. In the shallow marine environments of the early Paleozoic, salinity was variable. Tidal zones concentrated and diluted. Intertidal pools evaporated. An organism living at the interface of water and land experienced constant osmotic challenge. The movement of water across cellular membranes in response to changes in solute concentration. Too much salt outside and water leaves the cell. The cell shrivels and dies. Too little salt and water floods in. The cell swells and ruptures. The tardigrade lineage apparently solved this problem by developing the capacity to survive extreme dehydration, to allow water to leave the cell in a controlled way, under the management of the CAHS proteins and whatever other protective chemistry was evolving at the time, without catastrophic collapse. This is called osmobiosis, the ability to enter a dormant state in response to osmotic stress. What happened next is not evolution's plan, it is evolution's arithmetic, an organism that can survive complete dehydration, that can vitrify its cellular contents and wait indefinitely for water to return, has, as a structural consequence, also solved many of the problems posed by freezing, by radiation, and by the vacuum of space. Because all of these stresses share a common mechanism. They damage biological molecules through the action of reactive chemical species, particularly hydroxyl radicals. Radiation generates them. Freezing concentrates them. Desiccation allows oxygen-derived radicals to accumulate in the absence of the aqueous buffer that normally neutralizes them. The tardigrade's protective chemistry, the CAHS glass, the Dsup armor, the upregulated antioxidant enzymes, the DNA repair pathways that run at full capacity during recovery, addresses all of these threats through the same underlying biochemistry. Not because evolution anticipated space, because the chemistry that solves one extreme turns out to solve several others. The tardigrade did not become invincible on purpose. It became invincible as a side effect of learning how to live through dry season. In November 1983, a team from the Japanese Antarctic research expedition collected a sample of the Moss Bryum argenteum from Yukidori Valley on the coast of Dronning Maud Land in East Antarctica. The sample was stored in a laboratory freezer at -20 degrees Celsius, and there it sat for 30 years and 6 months. In May 2014, researchers at Japan's National Institute of Polar Research thawed the sample. They recovered 2 adult tardigrades and a separate egg from the defrosted material. They named the individuals SB1 and SB2, Sleeping Beauty 1 and Sleeping Beauty 2. They placed all 3 in individual wells on agar plates with algae as food at 15 degrees Celsius. SB1 began to move within days. It was slow at first. The recovery process took longer than normal, evidence of accumulated damage that needed repair. But it moved, and then it ate. And 23 days after thawing, it laid its first egg. It went on to lay 19 eggs. 14 hatched. The hatchlings were morphologically normal. They reproduced. SB2 showed initial movement. Its 4th pair of legs moved on the first day after rehydration, but never fully recovered. It died 20 days after thawing. Of the 3 individuals, 1 adult survived and thrived, 1 adult died, and 1 egg hatched into a tardigrade that lived a completely normal life. This is the record for tardigrade long-term survival. 30 years in a frozen state, followed by recovery and reproduction. It is an extraordinary result. It is also, in the context of what other organisms have managed, worth placing carefully. The previous record for any tardigrade, 9 years in dry storage, was already impressive. But the record for long-term cryptobiotic survival in other organisms extends considerably further. Rotifers, microscopic animals with certain biological similarities to tardigrades, have been revived from Siberian permafrost after approximately 24,000 years, confirmed by radiocarbon dating. Nematode worms have been revived from permafrost samples estimated at 46,000 years old. Plant seeds have germinated after tens of thousands of years in Arctic conditions. The tardigrade's 30-year record is genuinely remarkable. It is not the upper boundary of what cryptobiosis can achieve. This is not a criticism. It is a corrective to the mythology that has grown up around the animal. The tardigrade is extraordinary. It does not need to be falsely positioned as the supreme champion of every survival category to remain extraordinary. What the Sleeping Beauty experiment actually demonstrated, beyond the raw survival number, was that the recovery process involves significant, time-consuming repair of damage accumulated during dormancy. SB1's first egg took nearly twice as long to hatch as subsequent eggs from the same individual. The system does not emerge from 30 years of freezing undamaged. It emerges with an inventory of molecular lesions that must be fixed before normal function resumes. The tardigrade can survive the duration, but duration has a cost that it pays on waking. In 2007, tardigrades in their ton state were attached to the outside of the European Space Agency's Photon M3 satellite and exposed to open space for 10 days. They experienced hard vacuum, temperature swings from -272 to +150 degrees Celsius, cosmic ray bombardment, and unfiltered solar ultraviolet radiation, which, without the atmosphere's protective filter, is lethal to most unshielded biological material. When the satellite returned, the tardigrades in the toon state, protected from direct UV by a thin metallic filter, survived in significant numbers. Some that received full unfiltered UV exposure also survived, though at lower rates. The experiment was widely reported as proof that tardigrades could survive in space. This is technically accurate and somewhat misleading in its implication. The tardigrades survived in their ton state, a state of suspended animation in which they were not living in any active sense during the exposure. They were not metabolizing, not sensing, not responding. They were, for all biological purposes, inert matter with the structural properties of a very resilient glass. What the experiment demonstrated is that the ton state is sufficiently chemically stable to survive 10 days of space exposure. It did not demonstrate that tardigrades can survive in space as living animals. It demonstrated that their dormant chemistry is more stable than most biological chemistry. This distinction matters because it relocates the impressive thing. The impressive thing is not space survival. The impressive thing is the ton state itself, the fact that an animal can convert its living biochemistry into something so chemically inert that a week of vacuum and radiation leaves it recoverable. That is the feat. The space exposure is merely the test that revealed it. And that feat, again, was evolved in a tidal flat, not a vacuum chamber. Here is the irony at the center of tardigrade research. The wrong question. How does this animal survive the impossible? Led, eventually, to the right answers. Not because the question was well framed, but because the biology was generous enough that even the wrong frame revealed genuine mechanisms. The Dsup protein is now being engineered into nanoparticle delivery systems for cancer radiotherapy. The MIT study, published in February 2025 in Nature Biomedical Engineering, showed that Dsup mRNA delivered to oral or rectal tissues before radiation exposure reduced DNA damage in those tissues by approximately 50%, without protecting the tumor itself. The protein is expressed transiently. It appears, does its work, and degrades within hours, leaving no permanent foreign material. The CAHS proteins, the biological glass that holds tardigrade cells together during dehydration, are being studied as potential excipients for stabilizing biological materials during dry storage. Vaccines, protein therapeutics, and cell-based medicine all face the challenge that biological molecules are fragile and require cold chain refrigeration from manufacturing to administration. A protein-based glass that can suspend biological activity indefinitely at ambient temperature would represent a significant logistical breakthrough. In 2025, a bioRxiv preprint demonstrated that CAHS proteins can protect synthetic lipid membrane systems during dehydration and rehydration. A first step toward engineering tardigrade-inspired desiccation tolerance into biological assemblies that do not naturally possess it. Both of these applications arrived because researchers went looking for how an indestructible animal works, and found instead how a highly optimized chemistry of desiccation tolerance happens to solve radiation damage and dehydration instability as structural consequences. The tardigrade did not offer us a solution. We found one while asking about the wrong problem. Let us state the evolutionary paradox of the tardigrade as precisely as possible, because precision is what it deserves. Over approximately 530 million years, the tardigrade lineage developed a suite of molecular protections against osmotic stress. Proteins that vitrify under dehydration, antioxidant enzymes that neutralize reactive oxygen species, DNA repair pathways that run at full capacity after dormancy, and a DNA-binding protein that physically shields chromosomes from radical damage. All of these mechanisms were responses to conditions the animal actually encountered in its actual environment. As a structural consequence of this chemistry, The tardigrade is also protected against radiation doses that no organism in its environment ever generates, temperatures that do not exist in any biosphere on Earth. Pressures equivalent to the deep ocean floor even for moss-dwelling species, and the vacuum of interplanetary space, which no tardigrade has ever naturally encountered, and which has not existed in any environment the tardigrade lineage has inhabited in 530 million years. Evolution did not build these protections. Evolution built desiccation tolerance. The rest came along for free. This is what biologists mean when they call it over-engineering, though the word is technically wrong because engineering implies intentional design, and nothing about this was intended. What we are seeing is not engineering. It is the expansion of a survival toolkit to its logical extreme, where the toolkit turns out to address threats the animal will never naturally face because the underlying chemistry is so fundamental that it generalizes. A better word might be serendipity or surplus. The tardigrade built a house to withstand rain and accidentally built something that would survive a nuclear winter. There is a version of this story where the tardigrade is a miracle, a design beyond nature's usual limits, a demonstration that life can go anywhere, do anything, survive every conceivable extreme. That version is popular because it is satisfying. It turns a half-millimeter animal into a symbol of indomitability. I think that version underestimates it. The actual story of the tardigrade is stranger and more interesting than the miracle narrative. It is the story of what happens when a very simple animal solves a very specific problem so thoroughly that the solution overflows its original context. It is the story of how the chemistry of wet moss, the humblest, most overlooked ecosystem on the surface of the Earth, turns out to contain a set of molecular tools with implications that extend to cancer treatment, to space medicine, to the long-term preservation of biological materials. The tardigrade did not evolve to be useful to us. It did not evolve to be invincible. It evolved to handle the water cycle in a patch of vegetation on a rooftop, and it did this with such molecular precision that the solution generalized to problems that neither the tardigrade nor evolution could anticipate. We have been calling it the world's toughest animal for 250 years, and we were right about the toughness and wrong about why it matters. It matters not because it is tough, but because it is precisely engineered for a problem we had not thought carefully about. And that precision turns out to be readable and transferable and applicable in ways we are still discovering. Somewhere right now, a tardigrade is moving through a water film on a piece of moss, doing what it has been doing since before the dinosaurs, completely unaware that its proteins are being expressed in mouse tissue in a cancer research laboratory in Cambridge, Massachusetts. It would not care if it knew, but we should.